This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1227, recorded on the 13th of May, 2025, and it's Friday. Yep, Friday the 13th. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Madison, New Jersey, Brianne Barker.
Hi, it's great to be here. It is 79 degrees and cloudy here. Much cooler than earlier in the week, but still a good day to be inside podcasting.
27C and cloudy here in Chelsea, which I guess is a neighborhood of Manhattan. Also joining us from Western Massachusetts, Alan Dove.
Good to be here, which is a neighborhood of Massachusetts.
What, good to be here is?
Yeah, Western Massachusetts.
Yeah, it is.
It's 77 Fahrenheit, 25 C, and it's mostly cloudy, but very pleasant. Nice light breeze.
So these places like Chelsea and NoHo and Soho, are these neighborhoods?
Those are neighborhoods, yeah.
Yes.
It's very informal stuff, right? It has no taxonomic ranking.
Yeah, I mean,
No.
technically NoHo is north of Houston, Soho is south of Houston.
Yeah, I mean, you'll see some sort of street signs labeling things, especially for parking.
Yes.
Mm. So Chelsea is another one. Then we have Midtown, I guess, is one, right?
Yeah.
Upper East Side, Upper West Side, Harlem. Harlem is another one.
Washington Heights.
Right. Okay, these are neighborhoods.
I was never quite clear on where Washington Heights ended and Inwood began.
That's right.
But
There's
that's
an
another-
Inwood section. They call it the Inwood section of the Bronx.
Yes. Well, there's the Inwood section of Manhattan, too.
That's right. It spans both. Does Brooklyn have neighborhoods like that, too?
Oh, gosh,
yeah.
yes.
Oh, yeah.
Like Crown Heights and
Yeah?
Bensonhurst and
Isn't Green, is it
Bensonhurst
Green
is
Point
Queens.
or Greenbrook or
Greenpoint. Greenpoint.
Bedford-Stuyvesant. Is that Brooklyn? I think
I
that's
think
Brooklyn.
so. Bushwick is another one.
I was like, there's the trendy one, but what's...
But it
the
keeps moving.
trendy one
That's-
is
yeah.
the trendy one is it's the most expensive one. Brooklyn neighborhoods. It's not
For a
gonna
while, I
happen.
think it was the Dumbo neighborhood down under the Manhattan Bridge overpass.
Mm-hmm.
Gosh, Flatbush.
Yep.
Oh, there's a part of Brooklyn called Pigtown. Oh my gosh.
Oh.
Ocean Parkway, East New York, Bed-Stuy, Bushwick, Greenpoint. I have a friend from Brooklyn. He grew up in Greenpoint. He calls it Greenpert. Williamsburg.
Williamsburg, also one of the trendy ones.
What's the one I'm thinking about that's very expensive? I'm not seeing it. on this
I think
list.
a lot of these
Williamsburg.
neighborhoods we've named are pretty expensive.
Park Slope
Oh,
Park Slope.
yes.
Oh, yeah.
is the one I'm thinking of. Then we have Red Hook. That's a nice name for a neighborhood.
Yeah.
And of course, Coney Island would be one
Yeah.
this way. Sheepshead Bay, Plum Beach. The thing is, Brooklyn is divided into central, eastern, northern, northwestern, southern, southwestern Brooklyn. A lot of history here.
Yes.
Okay, that is our little diversion today. We
There will
started
be
with
no further
Chelsea.
digressions during the show.
No, definitely not.
We have digression police. Well, if you enjoy these digressions as well as the science that we give you, we'd love to have your support to produce these programs. We are a 501c3 nonprofit, so your contributions are federal U.S. tax deductible. And we give you science, real science. We often give you the literature, which is science, and no lies, no misinformation. So if you like that, want to learn more, microbe.tv/contribute. ASV 2025 is coming up. It's hosted by McGill University in Montreal, and it will be at the Palais des Congrès de Montreal, July 14th through the 17th. The website is asv.org/ASV2025. It's a month away. Start looking at the program online, plan your activities, work on your talk or poster. If you've attended ASV in the past, now you can look for your badge holder. Get one of your old badge holders and bring it so we don't have to give you a new one. Yeah, one of ASV's sustainability initiatives is to reuse the name badge holders. Put it in your suitcase. I did that once. The last time I went to an ASV, I used one from 2015.
Nice. Can you use a badge holder from someplace else entirely?
You know, in the spirit of sustainability, they should let you do
You
that.
probably could, but
Yeah.
the ASV ones are pretty nice. So
They are pretty nice badge holders, yes.
They're very big.
They have like a pocket.
They're
Yeah.
also called nerd flags.
Well, yeah.
Someone told me that at the Madison ASV not too long ago. They walked into a bar with their badge on. They didn't take it off, and the bartender says, "Why don't you take off your nerd flag?" Nerd flag.
Fly it proudly.
Standard registration for the meeting ends on June 30th. After that, the rates go up, so save some money. And also the reservation deadline for the Campus One MTL Residence Hall has been extended to June 12th. Sign up by adding the reservation to your meeting registration.
You might have to use your time machine since that was yesterday.
Huh.
Yes.
Well, fire up your FTL.
Well, just going faster than light probably won't get you back in time, though.
Won't get you back in time, no? It'll just stop time.
Well, yeah, you'd stop time when you hit light. Your time would continue to pass in your perspective, but then when
But the rest of our time would not.
the rest of our time would, you would appear to get someplace instantly from your perspective.
Okay. No, we're not going back. All right, let's take that out of the notes because we don't need that anymore. Okay, a little bit of news.
Can we take some of this news out of the notes too?
Yeah, can we just make it not happen?
Sure. You want to not do the news?
No, we can cover the news.
We can cover the news. We just want to make the news actually go away.
Yes.
Yeah, I agree. We used to have better news, right?
Yeah, not as bad. I'm not sure I would call it better.
Yeah, you're absolutely right. As soon as I said that, I
No, sometimes we had news like, look, a new antiviral, look, a new vaccine, look, a new thing.
This is a Science article by Jocelyn Kaiser. NIH terminates network aimed at stopping pandemics before they start. Yeah. So the administration has shut down these Centers for Research in Emerging Infectious Diseases, which were launched 5 years ago, and they collect and characterize mosquito-borne viruses and other pathogens that can go from animals to people. And they've said, no more. It's it. They said it's not a good use of taxpayer funding, which is, again, one of these typical administration statements that are just lies.
Yeah.
Right.
And some people believe them.
This is like telling the military to turn off all their radar. Right? Not a good use of taxpayer money. We don't need to know what's coming in or flying past or anything like that.
Could have all the air traffic control towers turn off their radar too.
Yeah, that'd be good too.
Sure,
Why do you
we
need
don't
to
need
bother to look for stuff that might happen? Hasn't happened yet.
One of the things they say here is that the research is unsafe. You shouldn't be collecting dangerous pathogens because you'll just bloody infect yourself. Well, that's a typical Republican position on research.
They do know that the people who were infected with them, who you're collecting from, are infected.
Yes, right.
Anyway, this is just the beginning. They'll do more. But this is not a lot of money. It's $83 million a year. And so that's not much for being aware of what might be down the road. And David Wang at WashU says the NIH is pandering to lab leak conspiracy theorists. And it's incredibly short-sighted. But that is the mantra of this administration, incredibly short-sighted.
And pandering.
Yeah. All right, now the next two are not any better. First, we have a CNN story. RFK Jr. removes all current members of the CDC Vaccine Advisory Committee. So we had 17 members of this committee, which, they advised CDC on what vaccines to give to who and when. And so the states can take this advice and use it.
This is
They
the
choose.
ACIP Committee, Advisory Committee on Immunization Practices.
Which used to contain Paul Offit.
Yes.
Well, he's actually on he used to be on ACIP, and then he also, right now he's on the FDA panel. The future of that is in question also. Who knows? But, the people on the committee traditionally are pediatricians, epidemiologists, immunologists, people who know their stuff.
Vaccinologist, maybe.
Yeah.
Yep. But Kennedy has just fired them all because he said they have conflicts of interest. But he said they're all paid by Big Pharma, but they're not. They did a review of the conflicts a few years ago, and there's no conflict with any of them. So many people are upset about this. And Paul Offit is quoted somewhere. Is it in this article? Here. No, it's Michael Osterholm. So Offit has said that basically they have lost the trust of the medical community. No one's going to listen to ACIP anymore.
Yeah.
Yeah.
Because
well, not only did he sack the entire ACIP as it was, he then appointed a whole bunch of anti-vax and other fringe folks.
Yep.
Pretty much.
Michael Osterholm said the firing of ACIP represents one of the darkest days in modern public health. And then, yes, as Alan said, we now have a new slate of advisors. I have a story from NPR by Will Stone and Pian Huang. RFK Jr. names new slate of vaccine advisors after purging the CDC panel. All of these individuals, he said, are committed to evidence-based medicine, gold standard science, and common sense, which is actually the opposite of what they're all committed to.
Yeah.
Yeah. I think one thing that to me was really telling was that these two moves happened a day apart. He removed the first set of members on Monday and added this other set on Tuesday. And on Monday night on social media, I saw someone post, we'll know, it's going to be bad if these people get appointed. And they sort of predicted who would be problematic appointees. And the majority of the people who were appointed were actually part of that list that people were making about who would be, telling that he was trying to go in that direction.
What's the worst you could do here?
Yes. The only thing that, and this is bad, that, there's something that made me feel okay. And the one thing that made me feel okay about it was that he didn't name Wakefield.
No. Okay. Yeah, that's true.
It would probably be very toxic.
Right.
But he has, as someone said, he did not pick people with strong current expertise in vaccines, but rather a committee skeptical of vaccines, possibly willing to implement an anti-vaccine agenda.
Yep.
Well, I mean, the ACIP is supposed to recommend vaccines, not recommend them. But Kuldorf, an epidemiologist and biostatistician, is one of the co-authors of the Great Barrington Declaration with Jay Bhattacharya, right? Remember that?
Yep.
No lockdowns, let it rip.
Mm-hmm.
He declined to comment when reached by NPR. Now, if you have something to hide, you decline to comment. If you think you're doing the right thing, why not comment? Then we have Malone, Robert Malone. Oh, my gosh. He's a guy, he worked on mRNA vaccines long ago. Pissed off he didn't get the Nobel Prize. And then turns around and starts spewing misinformation on the Rogan show. And now, because of that appearance, he's well known and he loves it. He's going to say no mRNA vaccines because they didn't give me credit, stupid things like that. And we have Vicky Pebsworth, who was on the board of the National Vaccine Information Center, which sounds legitimate, but it's not. It's an anti-vaccine information center.
Yeah, this whole list is like that.
It's horrible.
I found it particularly interesting that one of the members, Meissner, had been on the committee before and had left because of conflict of interest disclosures.
Yes.
And so that was the reason why the people had been removed the day before, and yet he was named.
Yeah.
So this is very bad, folks, and I think you're going to start seeing them not recommending vaccines that they think are bad. So RFK has already said that, for example, the HPV vaccine is the worst vaccine ever. It kills more people than it saves. And this is a lie. It does not.
No.
It does not. But he can just make these lies and nobody calls him out. Right? Or if you do call him out, he says, well, I didn't say that. But you did.
Was something that we did say is about how all of the studies that came out within the past year looking at the number of cancer deaths that the HPV vaccine saved.
Yes.
Amazing,
So
yes.
I would recommend the evidence on that side.
Yeah. Yeah, we have episodes about this.
So he would just say, oh, those are all conflicted studies, blah, 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 right?
He would.
So it gets worse every week, folks. And if you are worried about it, you should write.
Write to your congressmen and senators.
In fact, I have I have to add this to the letters. I have another letter. If you remember, a couple of weeks ago, an organization put up a website where you could easily write letters to your senators and representatives. Now there's one calling for RFK Jr.'s resignation. So I'm going to put that up while we're talking here. All right, we have some papers today for you. A little bit of virology for you today.
Yes, we do.
Alan, the first paper is Genomics Reveals Zoonotic and Sustained Human Mpox Spread in West Africa. It's an accepted paper, but it's still in the preprint form in Nature.
Yeah, and I wanted to comment on the whole thing has this watermark, accelerated article preview.
Yep.
And accelerated here, I highlighted the schedule of this. It was received by Nature on June 18th, 2024, accepted May 9th, 2025.
Yeah, that's
accelerated preview
yeah.
went online May 19th, 2025. So yes, they got it online fast, but it took a while for it to accelerate.
Yep. Anybody
Anyway,
have a summary they'd like to give?
I can do one if nobody else. Okay,
Yeah.
So first of all, the authors, we have 3 co-first authors, Edith Parker, Ifeanyi Oma, and Delia Doreen Juicy. And we have 2 co-senior authors, Richard Najum and Christian Happy. And the team is from Redeemers University in Osun State, Nigeria, Scripps in La Jolla, University of Edinburgh, Nnamdi Azikiwe University in Accra, Nigeria, Centre Pasteur du Cameroun in Cameroon, University of California, Los Angeles, Broad Institute of MIT and Harvard in Cambridge, Theogen Genomics, that's Cambridge, Massachusetts, by the way, Theogen Genomics, Highland Ranch, Colorado, Ministry of Public Health in Yaoundé, Cameroon, Nigeria Center for Disease Control and Prevention in Abuja, Nigeria, Africa Centers for Disease Control and Prevention in Addis Ababa, Ethiopia, Riga Institute at KU Leuven in Belgium, and the Harvard School of Public Health in Boston. And neither of the papers today, by the way, is open access, so apologies. You can read the abstract, and that's about it.
And listen to us.
And you can listen to us. So, quick summary of this. So this paper is about Mpox, the virus formerly known as monkeypox, which has been around for a long time. It has typically been a sporadic zoonotic infection. Somebody gets it, they recover or not, and then that's that. And then it happens again a few years later, something like that. But for some reason, it popped up in Nigeria and Cameroon after not being there for a long time. It showed up in 2017 and 2018. Then it spread worldwide in 2022. People probably remember that. We have a few TWiV episodes on it. That Mpox strain that went around the world is clade 2b lineage B.1, and it has several mutations compared to other clade 2b viruses. Seems to spread better human to human than other Mpox strains. But where did it come from? So this team, to try and figure that out, they sequenced more than 100 Mpox virus genomes that have been isolated in Nigeria and Cameroon from 2018 to 2023. They found 2 separate zoonotic lineages of the virus circulating in the border area and across the border between Nigeria and Cameroon, probably from animals moving through that area back and forth. And animals are notoriously bad about checking in and showing their passports.
I know. Can't believe it.
Yeah, so they found that the ancestor of the strain that became an epidemic probably circulated in southern Nigeria in 2013, emerged in humans around 2014, and spread undetected for a few years after that before being noticed as an outbreak in Nigeria. At least that's what the data are consistent with. So it now seems to be continuing to circulate in Nigeria through human-to-human transmission, but recent cases in Cameroon seem to be coming from repeated zoonoses. The importance of this work is it highlights how circulation within animals can carry a virus back and forth across national borders, and also how repeated human zoonoses can eventually lead to evolution of a virus that can sustain human-to-human transmission and go everywhere.
All right. Thank you, Alan.
Sure.
Mpox, a zoonotic disease, orthopox virus, poxvirus family, MPXV. It's a zoonosis, so that means it comes from an animal, a non-human animal, to humans, but we don't know what the animal is actually. There are
Which
two
is
clades.
one of the reasons it was changed from monkeypox to Mpox.
Yeah.
Right. And when something is zoonotic, this may be obvious to some listeners, but I know that it's come up with some of my students as a point of confusion, so I'll just mention it. It could be a situation where every human who is infected was infected from some animal. That's zoonotic. Or it can be a situation where we went from animal to human and then more human-to-human cases.
Give us an example of both, Brianne.
Okay. So one example that might be where we go from animal to human might be rabies. We could also think about something like West Nile, where humans are sort of a dead end. Every human who is infected got infected from an animal. Or we could think about something like the most classic might be influenza, but you could think of lots and lots of examples where we have an animal to human transmission and then sustained human to human transmissions after that. That would still be zoonotic infection. And as Alan sort of talked about really well here, those two patterns may be sort of some of the difference that might be seen in what's going on in Nigeria and Cameroon. And I've had students, like I said, who've been confused about that point when I talked about a zoonotic infection, that they think it means every person must have gotten it from an animal. And that's not necessarily true. And this sort of points that out very well in distinguishing these two different options.
And I think there's some kind of maybe an arbitrary point where we stop considering it zoonotic if it's transmitting well from human to human and all or most of its transmission seems to be human to human.
Yeah.
Then we just say it's
a human virus.
It's a virus. It's an outbreak.
I like zoonotic, the word. It's a good word.
I do too. And I think that arbitrary distinction that Alan mentioned is sort of an interesting one to think about, because sometimes there are things like this percentage of all emerging infectious diseases are zoonotic, or this percent of infectious diseases are zoonotic. And I'm like, but didn't all of these really come from an animal at some point in time? Maybe even before.
We came from animals.
Exactly.
How far back do we want to go?
Exactly.
The zoonosis is best for those viruses that for every human infection, it's a spillover. That's a zoonotic infection. But for SARS-CoV-2, as soon as it got into people, it was transmitting among people. It's no longer zoonotic, right? Because there are no animals involved.
No, but I think it is still referred to as a zoonotic virus because it originated in an animal.
Well, it depends who you talk to.
Right.
I will not, because it's no longer, it originated as zoonosis, absolutely, but it's now a human virus. That's how I put it.
And I think at the point where we are giving the virus then to other animals that didn't have it originally, we can stop calling it a zoonotic virus, because now it's a reverse zoonotic virus.
Yeah.
We own it.
Yeah.
Two clades of MPXV, and the first one has a typo. It was historically Googled after.
Yes.
Mm-hmm.
I think they were Googling something and they got it mixed up.
Clade 1 was historically Googled off restricted to animal populations.
So funny. Yes, it is an accelerated article. So Clade 1 in animal populations in Central Africa. And I have my map here just so I get it all right. And Clade 2 to Western Africa. And that's divided into subclades 2A and 2B. first identified in humans in 1970 to 2017. They were not very common, these human cases.
And
Mostly
to be clear, Cameroon and Nigeria, which we're going to be talking about here, are West Africa.
They're West Africa. They actually have a coastline, and Nigeria shares a long border with Cameroon. So you think of the part of Africa where it bends around. They get that western bulge, and then it curves around and starts to go down. That's where Nigeria and Cameroon are located. And of course, Cameroon, very famous because that's where SIV spilled over into people and became HIV. Cameroon. Never been. Have you been, Brianne, to any of these places?
I have not, and I so want to.
Would like to go. Okay, so these were very rare, mostly zoonotic and limited human-to-human transmission. But, of course, we have seen in recent years a large increase in cases, right, and all over the world, both in these countries, in the endemic and the non-endemic countries. So in Nigeria and Cameroon reported their first cases in over 3 decades in 2017 and 2018. So those two countries.
This is just to, we usually introduce the virus itself in case it wasn't obvious. Mpox is a pox virus. It's a large double-stranded DNA virus with an envelope.
Yeah, I was hoping Rich Condit would be here.
Yes.
But rather than educating the world, he's off doing something else.
Oh.
Yeah, I saw in May 2022, a clade 2B lineage called B.1 started to travel around the world, causing multi-country Mpox outbreaks. And those are, you have heard of those most certainly. And this looked like human-to-human transmission. So people thought, oh, maybe this is a new way that the virus is transmitting. And this B.1 lineage was diverged from the clade 2B genome that was found in Nigeria in 2018. It has a lot of mutations in the genome. This will become important in a bit. Mutations in the genome characterized by being caused by an enzyme in the host cell, which is a cytosine deaminase called ApoBec3. And it's a host antiviral protein that deaminates cytosines, and as a consequence, you get mutations in the genome, and it renders them non-infectious. And so this new outbreak was characterized by mutations that look like they were caused. It's a signature of ApoBec. And you don't see this ApoBec signature in zoonotic infections. And so their hypothesis, or the hypothesis in the field, is that this genomic editing by ApoBec3, right, because it's taking the amino group off of cytosine, and then
that turns it into uracil.
Turns it into a uracil, thank you. And then when the DNA is copied, of course, or the RNA is copied, now a uracil, it goes to an A instead of the original C, which would go to a G. So you have a mutation.
Or if this is in the DNA, since we're in a DNA virus, having a uracil in the midst of DNA basically triggers repair proteins and excision of that base. So
just remember that ApoBec genomic editing is thought to be a feature of human-to-human transmission, which is a nice marker to have because we don't always have that. Okay, so that's the idea here, that human-to-human transmission in Nigeria is driven by this. And in Nigeria, by the way, monkeypox, MPXV emerged in 2016. It circulated and diversified into different lineages. But in contrast, Cameroon recent incidents is most likely driven by zoonotic transmission, sporadic cases here and there, not sustained human-to-human transmission as we saw in Nigeria. And now one thing that you need to know is that the area around the border of Nigeria and Cameroon is the Cameroonian Highlands Forest Belt. It's a forest, and guess what lives there? Animals. So there are plenty of suitable animal reservoirs for this virus. We don't know which one. In addition, there's agriculture, there's hunting, there's human settlements, all at this human-animal interface, which helps transmission as well. They call it a porous border between Nigeria in Cameroon.
Yeah, so this area is mountainous jungle.
Yep.
And that's pretty hard to patrol what's going on, and lots of animals.
Yeah, so animals, they can give their viruses to people. The people move across the border. They can carry the viruses with them. So lots of opportunities for spread.
Animals probably are not staying in one country or the other.
No.
They don't get a border check, huh?
No, bats are notorious about just ignoring that stuff. Very bad with paperwork.
You know, this brings to mind, we did a paper on TWiV yesterday where they were talking about using bacteria for bioremediation. So if there's a spill, right, you add. But then they said at the end, we have to be careful about using bacteria for forced labor. And they were totally serious about it.
Wow.
I've never heard such a thing. I mean, I can understand a mouse or a beast of burden, right?
Even that's a stretch. I mean, if you're going to start according rights to bacteria, then we're done.
Doesn't that mean that like all of recombinant technology?
Yeah, you can't do any more research in using E. coli, Brienne.
It also means that antibiotics are genocide. So, I mean, things are not going to go well if we sign on to this.
I just thought it was very interesting. Anyway, back to the paper. So what they did here, there's a lot of questions about where transmission is occurring. Is it human to human? Is it zoonotic? And we don't have enough genome sequences to answer these questions. So what they did here is to establish a Pan-African consortium that would put together a big database of MPXV sequences, genome sequences. Okay? And so that's why we have so many people on the paper authoring it, because they probably all contributed samples. So they got 118 near-complete MPXV genomes, 109 from Nigeria and 9 from Cameroon, sampled between 2018 and 2023. Now, the Cameroon number is small, and they admit that. And, you know, it's hard to get samples from various places. But they're working on it. This continues. Right?
And a major limitation here is that neither of these countries has the infrastructure to go out and collect a whole lot of samples.
Yeah, that's right.
Right. You know, fortunately, when you think about the affiliations, we noticed that one of them was the Nigeria Center for Disease Control. Yeah. But there is not a Center for Disease Control in every country, not, for example, in Cameroon. And so, you know, that makes getting all of these samples really challenging.
And this is the kind of project, this is the kind of effort that is only possible in that area with foreign aid, particularly from organizations. I don't know if USAID was directly involved in this, but certainly, you know, grants from overseas, especially from the United States, are driving this research.
Someone search the document for USAID.
I'm going to the end to the acknowledgements to see if it's mentioned.
Anyway, they know where each of these genomes comes from, and all of them are clade 2B, right? That one that has been spreading most recently, like we discussed before. Okay, 118 near-complete genomes. They look at their phylogeny compared to other clade 2B sequences, and you can see that the new sequences, the new Nigerian sequences, are interspersed through the lineages on the existing phylogenetic tree. And they say this suggests that the virus has cryptically circulated and diversified, largely driven by APOBEC3 in the human population in Nigeria for a prolonged period after it spilled over. So you can tell that from the phylogenetic tree.
So no USAID,
right?
Yeah. But Flu Lab, TED, multiple NIH grants, World Bank, many foundations, many Wellcome Trust, Rockefeller Foundation, African CDC, European Union. So a lot of funding from a lot of organizations.
And 7 of the grants are from NIAID.
Yeah.
Oh boy. All right. So this part, the conclusion
I wanted to mention that for no particular reason.
No,
I can't imagine why.
Yeah. For folks that this budget is being cut severely, NIAID and NIH, as you know. All right, so this means that the primary driver of Mpox cases in Nigeria is sustained human transmission. You can tell that from the genome sequence analysis, which is very cool. Now, in contrast, you have 9 Cameroonian sequences, and it's coming from a different branch on the tree, and it looks like those are the result of zoonotic transmission because they're not interspersed with the human sequences.
So they really are coming from a branch on a tree, probably.
They are coming from a branch on a tree. So if you look at the APOBEC signature, which we talked about, the Nigerian cases, they look at this and they say the Nigerian cases clearly have signatures of APOBEC genome editing. So that shows that the Nigerian sequences are the result of sustained human transmission. But in the Cameroonian, there are very few APOBEC type mutations. And so that's another support for the idea that the Cameroonian sequences and a couple from Nigeria that don't have the APOBEC signature are all zoonotic transmission. In fact, all of the sampled Cameroonian cases were zoonotic. So this is interesting because this is happening at the same time. You have human-to-human transmission, and then in a neighboring country, you have zoonotic transmission continuing. It's very interesting, right? It's probably happening also with HIV, but we don't know it. There's probably lots of spillovers, but they never get anywhere.
Right. And there probably have been for a long time.
Yes.
All right. So as Alan said, there's the border of Nigeria and Cameroon is this complicated forest belt that goes into both countries. So the forest grew over the border. It doesn't care about the border, right?
The forest predates the border.
Yes, right.
I'm pretty sure.
Yeah, well, there's actually,
Same.
there's no line.
Yeah.
It's just we put it on a map. So a lot of animals in here that could in principle be hosts of MPXV. There's hunting, wild game trade, human movement, as we've said. And so they wanted to try and figure out if they could see cross-border zoonotic transmission between the countries, right? And in fact, 2 of the 4 Nigerian zoonotic sequences cluster with Cameroonian sequences. So that suggests they came across the border, those viruses, right? Originated in Cameroon, were brought across the border, and the sequence can show that they're distinctly different from the Nigerian sequences. There's also a new lineage that they find here from a part of Nigeria, which it seems to represent independent zoonotic transmission that diverged for a long period of time in an animal population. You can learn a lot from sequences. It's very cool. So when did this happen? How about the dates? Can we get some dates out of this? So yeah, you can. You can apply Bayesian phylogenetic reconstruction, which you can do in your own home. It's not dangerous. People say, don't try this at home. Well, you could do this at home.
You could do this at home. If you have the sequence data, you can do all of
the
the
sequence
data.
and some statistics.
Yeah,
Yep.
It's not that bad. So to see roughly when the cross-border dissemination occurred, right? And so they find this Cameroonian lineage and another lineage had a common ancestor that were circulating around February 1966. It was 13 years old, and this virus is circulating still. I didn't know that, right? And further analysis, basically,
I didn't exist.
You didn't exist, neither
Nope.
did Brianne. That's for sure.
Mm-mm.
That's for sure. Oh boy! So these sequences suggest that, or these kinds of analyses, these Bayesian analyses, suggest both recent and older spread across. They call it a porous border, likely originating in an animal population that had quite a lot of diversity in the viruses, the Mpox viruses that were in them. All right, so what is the zoonotic ancestor of the human MPXV? And they have a lot of genomic data to look at, and they find a single zoonotic origin for the virus that is currently being transmitted among people in Nigeria. So that's very cool. There's a sister lineage to HMPX1 called ZX, and these sequences have a share a common ancestor with HMPXV1. So that's the closest zoonotic outgroup to HMPXV1. So basically, this outgroup of ZX-related viruses have a common ancestor that circulated in animals in late November 2013, not too long ago, right? And they estimate that the transition to sustained human transmission, right, from the zoonotic spillovers to sustained human transmission, which we see in Nigeria, was about August 2014. That's pretty precise, right? Of course, there's an interval. There's a wide interval, but that's about the average. So the time to the most recent common ancestor of HMPXV1 is August 2015, and that's about the time when it started to diversify in humans. Now, you may ask, and it would be a good question, why? And we don't know what happened back then. It's a lot of chance events that have to occur. It has to get into a human. It has to be transported where there are more humans. And, you can say, why 2019 did SARS-CoV-2 emerge? We don't know.
It finally the right ingredients came together.
Yeah,
It's a perfect storm.
I think that was one thing I found really interesting in this paper is they almost strenuously avoid naming any organism other than humans. So there's no hypothesizing about what sort of other hosts might be involved at all. It's just sort of spillover from some forest host, and they don't say anything about what that could be.
Do you think they do that on purpose, Brian?
I'm impressed by how cleanly they manage to not mention it anywhere.
Yeah.
It is very tempting to speculate, but
right.
Again, this is mountainous rainforest. What couldn't it be? There's all sorts of stuff in there, and odds are good that if they mentioned something in the paper and said, maybe this came from a bat, well, we could soon find out they're wrong.
Right. So I sort of almost wonder if, at some point there might be a future work trying to describe what this, perhaps small mammal rodent, for example, might be. But you might imagine maybe there's something in 1966 that happened that changed what was going on with the population of that organism. Maybe there was some kind of change in the forest at that time. Maybe there was some additional change in the virus that led to more human adaptation later. Those are things that maybe we could start to find out now that we have some of this information and can start to look at some of those types of questions.
And the late 60s were a major transitional period in many African countries as European powers pulled in, pulled out, rearranged, changed things, and, local national governments took charge, and stuff changed a lot.
Yeah.
So I'm pretty sure that something on the human side changed and maybe laid the groundwork for this unknowingly.
So they conclude that the precursor lineage of HMPXV1 circulated in some animal in southern Nigeria for about 50 years. So it came over the border from Cameroon and circulated in southern Nigeria for a long time before it emerged and got noticed, right? And, it got noticed because there were cases. So they say this was cryptic circulation before that for quite a while in these forested border regions in the south region of Nigeria. And in fact, these southern states of Nigeria were the early epicenter of the Mpox epidemic. And the northern states only started to have significant numbers of cases after the 2022 resurgence. So that was the initial outbreak, and then it kind of quieted down, and then you had a resurgence in 2022. They note that there's a particular part in the South called the Rivers State, which is the primary source of viral exports, Mpox viral exports throughout the epidemic. 75 viral introductions originating in rivers, which they can, again, glean from their sequences. So they want to next know how widespread the viral transmission was by the time the outbreak was declared, which was in September 2017. So that's when there are a lot of cases, and then people say, oh, we're having a Mpox outbreak. So they look at the sequences that they have and where they are, right? Where the sampling happened. So this is called phylogeographic reconstruction. And they find that the virus had spread over 500 kilometers beyond the Rivers state, right, which is exporting all this stuff into other states before the first case was detected, which was September 2017. So just to summarize this, the epidemic begins in Rivers state, spread of the virus to the south and southeast states and Lagos, and then outbreak declaration, and then delayed dispersal to the north. And it's just sequences, folks. It's very cool. Well, geography, sequences, a little bit of epidemiology as well. And they also want to know whether, so now that, there's these persistent transmissions in Nigeria, human-to-human transmission. Are those continuously being seeded from River State, or were there locally persistent transmission change? So they do a lot of analysis of the data to get at that question. And they conclude that the River State was the persistent source, and the local epidemics were largely driven by repeat introductions. All you wanted to know about Mpox. Now, what drove this pattern, right? Why did this happen? And so they try and look at other factors that might influence the movement of this virus from a zoonotic to a human. They look at epidemiological, demographic, geographic variables, economic variables. And the only thing they can come up with is the main predictor of virus dispersal is that it originated in the River State, which we know.
Yeah.
And population density was positively associated with dispersal. But there's nothing in particular of what they looked at that correlates with dispersal. Like they probably looked at hunting practices or food transport. None of that.
Agriculture, economics. Yeah.
Yeah. None of that corresponds.
Nothing obviously correlates. Just the more people you have in an area, the more likely you are to spread it in that area. And it started in Rivers State.
So that's the story, folks. So you have this virus, zoonotic transmission in Cameroon. It is in some animal reservoir at the Cameroon-Nigerian border. It gets into Nigeria. It circulates cryptically, human to human, for 10 years, and then it causes an outbreak. So why does it cause the outbreak? They don't know, but they say that the transmission may have occurred in a more interconnected mobile population with connectivity to urban areas and transmission driven by behavior or demographic factors, right? So the idea is that it could circulate cryptically in a very, very rural population, not very dense, right? Sparse populations. But then as soon as it gets into someone who goes to a big city, it moves around, then boom, that could really explain it. But they don't know.
Truck drivers, traveling missionaries, anybody who moves around a lot.
Yeah. So basically they say we need to have more surveillance of the wildlife population to see what's going on here. And I hope they can get the money to do that. It's a lot of it is dependent on U.S. funding, which is drying up. And so I hope other countries can step up and help out. They also say that, this can help us with public health interventions to understand the patterns of transmission, except controlling these epidemics in Africa is difficult, right? It's not, we don't have equitable access to diagnostics, vaccines, and therapeutics. And they close by saying the global community can no longer afford to neglect Mpox in Africa or perpetuate inequities in therapeutic access in our vulnerably connected world. This is the thing, folks.
True story.
Yeah.
This is a connected world, okay? If you
no matter how rich and privileged you think you are, the problems of the poor will be your problems.
Yeah, I mean, we saw Mpox come to the U.S.
Yep.
already. And There are more outbreaks in Africa. There are going to be more here. And you cannot just say, we're going to isolate ourselves and take away all the money we're giving you. No, it doesn't work. It's a connected world, folks. And the people in charge don't get this.
Yeah.
They have this view of the U.S., which is an abomination, essentially.
Does not make any sense.
No.
Moronic. Morons have overtaken the U.S. We have to get it back. Okay. They have a second paper, which is in Nature, Microbiology. Now, this is a nice paper, which you can, there are no typos, and you can read.
This is it's been through the copy editing process, and
Breadth of influenza A antibody cross-reactivity varies by virus isolation, interval, and subtype. Alan, you want to summarize that?
Sure. So, single first author, which is a noteworthy thing these days, is Bingyi Yang, and the single senior author is Benjamin Cowling, and they are from, I actually bookmarked where I needed to scroll in this because of putting all the affiliations at the end, which I am still going to complain about. The team is from University of Hong Kong, University of Chicago, Hong Kong Science and Technology Park, La Jolla Institute for Immunology, University of California, San Diego in La Jolla, Johns Hopkins University in Baltimore, Monash University in Clayton, Australia, and UCLA. And my quick summary of it, which I hope I won't butcher too much of the immunology here. All right, so the basic question they're looking at is, what makes some flu seasons so much worse than others? We all know this is a phenomenon that occurs. The biggest reason is population immunity. How much people's flu exposures, whether infection or vaccination, generate immune responses that can protect them against the current year's strain of influenza virus. But we're not very good at predicting that. So this paper looks at the immune responses. This is an informatics data mining paper. They're taking published results from other groups and combining them and analyzing all the data.
Today is Statistics Day.
Today is
These two
Statistics
papers.
Days.
It is.
Yes. First, they look at immune responses in ferrets. So they're going to look at immune responses in ferrets and humans against various flu strains that have circulated over the past several decades, to see what drives cross-reactive responses from one year's strain to another year's strain. So first, they mined the data from studies where researchers infected ferrets with one flu virus strain and then tested which other strains the animal's antibodies could neutralize. That showed that viruses that emerged in years that are closer to each other generated more cross-reactivity against each other than viruses that emerged in years further apart. It's suggesting that immunological memory has a sort of a window time frame of a few years. So then they did a bunch of additional analyses to try to get to the mechanism behind that. But then they looked at humans. First, they looked at antibody responses in 6 infants who had documented influenza infections from 1995 to 2004. The reason they went with infants is because they were looking for infections where the kids had just gotten their first flu infection. And then looked at what viruses those cross-reacted with. So it's like the ferret experiment. And they found it gave similar results where it was adjacent years to where these kids were infected with the original virus were most likely to have cross-reaction with the virus they were first infected with. Then they went to a much bigger human data set, and this data set is across a wide age range. So now we're looking at people who have a more complex immunological environment because they've had multiple flu infections, most likely. Looking at those antisera, they found some evidence for original antigenic sin, which is something we've talked about before, and we're going to be talking about it again on this episode, where the first infection kind of sets the tone for the rest of the immune responses. But they also saw some similarities with the ferret and infant data where the closer you are to your earlier or whatever infection, the closer the year of isolation is, the more likely you are to have cross-reactivity. And they found that the immune response of the two main viruses they're looking at here it's influenza A, H3N2, and influenza A, H1N1, those two different strains. And they found the immune response to H3N2 seems to be narrower than the one against H1N1. Is that a good way to put it?
Yeah, I think so.
So that's consistent with population immunity maybe declining faster as H3N2 drifts, because it can drift more easily out of the range of population immunity compared to H1N1. The importance of this is it points us toward trends in population immunity against different flu strains, which could help us inform vaccination strategies and vaccine development strategies.
Okay, thank you very much. So influenza, seasonal influenza, not pandemic, over 100,000 deaths each year. Deaths, folks, even though we have vaccines and pre-existing immunity. So we need to better understand what's going on. People have different immune histories. There are different kinds of influenza viruses circulating. And of course, we don't know much about, as they say, the cross-reactivity conferred by earlier infections. So the titers of antibodies against it, and again here, we're focusing on antibodies, which are considered to be a correlate of protection. But of course, T cells are important also, but we're not going to consider that here. Antibodies can go down in titer over time, but the first virus the influenza virus that you're infected with, often immunity can persist for decades. Okay, so that's great, except the virus changes over those decades. And so that could be a problem depending on whether there's cross-reactivity between the antibodies that you first make and then the subsequent viruses. And that's one of the things that they're going to look at in this paper. Now, they compare it with measles virus, right, which is a virus that it's relatively antigenically stable. And antibodies against measles virus they say they have the half-life longer than the human lifespan. Oh my gosh. If humans live to 80 years old, that means you have measles antibodies your whole life. And the virus doesn't change. So it's good. That's why if you got measles in the 50s, you're still protected now. And they say you don't need to get vaccinated.
Or if you got measles vaccine in the 70s.
Yes. Why can't all viruses be that way, Brianne?
I think that's a virus mutation genetics problem, not an immunology problem.
Yeah, it is. So the viruses change. With measles and polio virus, we've used the same polio vaccine forever, and the virus doesn't change sufficiently to evade it. So that's another question besides what they're going to do here in this paper. But the point is that viruses like influenza and SARS-CoV-2 do vary antigenically. And so even though you may have antibodies to the first virus you were infected with that lasts your whole life, doesn't do any good at some point because the viruses have changed.
I guess it's technically like a plasticity of structural biology problem.
Yes.
Yeah, we talked about that on TWiV a number of times, how the structure of the virus limits what can change.
Yeah. Yeah, and in fact, SARS-CoV-2 and flu are good examples of this. We don't really know what SARS-CoV-2 is fully capable of yet because it's only been like 5 years. But what we see is that it seems to increasingly be exploring a little bit of territory that gives it just enough to propagate to the next season. Yeah. And that's kind of what we see with the other common cold coronaviruses. They've got an antigenic neighborhood that they frequent. And if you've got some antibodies against that, you've got some protection against that. Whereas flu can drift way, way off, and it can also shift. It can take segments from other influenza viruses. But what we're talking about in this paper is drift. And it has a wide latitude to do that can go potentially way outside what your immunity is.
So in this paper, they want to look at antibody cross-reactivity after a single infection with either H3N2 or H1N1 in both humans and ferrets. And as Alan said, they're using pre-existing data. So they don't do any wet experiments in this paper. It's all computational. And so remember, H3N2 influenza viruses emerged in 1968. And H1N1 in 2009. And when they emerged, they both caused pandemics. So they have, what they do is they look through the literature and get all these data on previous antigenic studies and reports on H3N2 viruses and H1N1 viruses. So for H3N2, it's from 68 to 2022, 102,000 titrations from 306 immunizations and 6,959 tested viruses. And for H1N1, 94,000 titrations, 60 immunizations, 6,900 tested viruses.
And I'm pretty sure that a major reason they looked at H1N1 and H3N2 and looked at antibodies and not T cells is because these are the things everybody has studied and these are the experiments everybody has done, so you can get large quantities of these
absolutely.
data.
I don't think that you could, well, I know for sure, you wouldn't go back to a paper from the 60s and expect to see the same kind of T cell study that you would see later on. And you wouldn't really be able to have, comparable data that you could put together in this way. Yep. So, they're specifically looking at HAI here because that's something that has been consistently used all of this time.
And you probably couldn't get enough data to look at influenza B.
Right.
So what they had, these data are generated in the following way. They take a ferret. There's one on the shelf behind me. You can see. And they inoculate it with a specific virus, just one, and then they let it make antibodies for a while, and then they take out serum, and they test it against a panel of viruses for hemagglutinin inhibition. Okay, so hemagglutination happens when you add influenza viruses to red blood cells. They make them clump, and you get a nice sheet formed in a 96 well plate, and if there's no hemagglutination, the red blood cells fall to the bottom, make a little pellet. And you can add antibodies to that reaction and block hemagglutination. So that's called hemagglutination inhibition, or HAI. And so the first time they look at this, they collect each ferret serum in these other studies as a function of the years when each tested virus was first isolated, right? So you have the year of isolation of the virus, and then they have serum before and after its isolation. And so they align all these data, and what they see in these curves is that HAI cross-reactivity. So they're looking for cross-reactivity between those initial antibodies and reactivity with the other viruses they're testing against. HAI cross-reactivity after a single infection mainly depends on the isolation time between the tested and the immunizing viruses, right? So for H3N2, they looked at 306 different H3N2s. There's this bell-shaped curve between cross-reactivity and the difference in isolation time. And so if the isolates that are tested against the immunizing virus were close in time to when that immunizing virus circulated, you get really good cross-reactivity. And the more you move away from that isolation point, the cross-reactivity goes down.
So if you had a virus, if you inoculated the ferret initially with a virus that was isolated in 2000, that was the circulating strain in 2000, and then you compare their antibody response to a strain in 2000 that was isolated in 2001 or 1999, it's pretty strong. But if you compare it to a 1968 or a 2016, it's much weaker. And those viruses out on the tails are much more likely to be able to get around immunity, weasel out of it, if you will, in the ferret.
Yeah,
they ferret out of it.
That's good. Yeah. And this is sort of, in some ways, totally expected, and in some ways, not expected.
Yes,
I guess is what I would say, because I think if you had asked me about this without, thinking about the data or without showing me any of the data, I might say, well, there's probably some structural feature that is related to cross-reactivity. Maybe if you have a particularly large HA or a small HA, maybe a large HA allows more cross-reactivity, or maybe an HA with more glycines, so tiny amino acids that aren't so bulky, or whatever. Maybe there would have been some structural feature. And if there was some structural feature, you would see that things with that feature sort of diverged from this pattern. They wouldn't follow this nice time pattern as well. But it really looks like the only thing that matters here is time. The only thing that matters is sort of how many point mutations, how much drift has gone on since you were originally infected, or since the virus you were originally infected with. And like I said, in some ways it's like, well, of course, it's just about how long it's been and how many mutations it's picked up, but also there's no other thing that seems to be really driving this cross-reactivity.
Yeah. Yeah, and it's interesting that, you think of genetic drift evolutionarily is the virus's response to population immunity, and it has to get around that by drifting enough that people aren't immune to it anymore. So in some senses, it's kind of surprising to see that the following year's flu virus isn't that different. I mean, you would have cross-reactivity, like it's not escaping by much.
Yeah, I mean, I guess that also tells you that the immune system has done a pretty good job of evolving to hit epitopes that are somewhat conserved.
And the horse you rode in on, like everything in that area.
So this is basically what they do to see if they should change the vaccine every year. They take, the antibodies that people have, and they react them against whatever is circulating, and they look at and there's a cutoff of HAI titer that is, we have to change the vaccine this year. So you are looking at that in a practical sense. So this is interesting for H3N2. 6 years after the immunizing virus was circulating, you have a significant drop in neutralization HAI titer. It goes down, but for H1N1 viruses, it's different. It's not such a peaked bell curve. It's broader. At 6 years, it hardly drops. The HAI activity hardly drops. So that's a fundamental difference between H3N2 and H1N1. So they say this shows a connection between the breadth of measured HAI antibody and the difference in isolation time between viruses, and there's something to do with subtype in this pattern, because we see the difference between H3N2 and H1N1. So that was new to me. I didn't know this. So then they next ask, how long does a 50% protection against hemagglutination, and so HAI titer of 50%, how long does it last for H1N1 versus H3N2 viruses? And so they grow and immunize H3N2. Well, not they, but others have done this. And they do titers in the same year. They have good titers above 40 in the same year. And then 6 years later, it drops to like 18. And 6 years later for H3N2 viruses. So the titer, so we're not looking at cross-reactivity here. We're just looking at the titer of HAI antibodies over years in a single immunized animal. For H1N1, these antibodies remain very high, regardless of the difference in isolation time. So again, another difference between H3N2 and H1N1. All right, so now we've been working so far without thinking about mutations, because now we know the mutation rate is high in influenza viruses, and that leads to amino acid changes in the hemagglutinin, which can lead to evasion of antibody binding. So they did a study where they look at the number of amino acid substitutions in the HA1 segment, which will mediate the resistance to antibody, and the associations between antibody breadth and isolation time that we've just been talking about. And so they note that the smallest amino acid substitution differences in viruses isolated in the same year is about 4 for H1N1. For H3N2, viruses isolated 6 years apart have a median of 17 changes for H3N2 and 11 changes for H1N1. So I didn't give you all the data. For virus in the same year, there are 4 median differences for H3N2 and 3 for H1N1. So within the same year as that immunizing virus, 4 or 3 amino acid changes. But for 6 years, you got 17 for H3N2 and 11 for H1N1. So that's different, right? It's really different. So that may partially explain the difference that we've seen so far between antibodies against H1N1 and H3N2.
Right. So H3N2 is changing more.
It is changing more.
Which makes it harder for antibodies to be able to cross-react.
Right.
Yeah.
But interestingly, the amino acid changes alone do not fully explain the differences in cross-reactivity with isolation time that they've previously seen. We got to put the isolation time and the amino acid changes together, and then that explains the data that they have seen where, you get this bell-shaped curve and so forth. So something is happening other than the amino acid substitutions, and we don't know what that is. They do talk about it later, but frankly, I didn't understand their explanation.
Yeah.
Maybe somebody
Good.
else
I'm
did.
glad I'm not the only one.
Nope.
Maybe
Okay.
someone else. No, okay. We're not going to be able to tell you why that might happen. I don't actually know why, but their explanation to me is obscure, let's say. But we do know that the more amino acid substitutions between the test virus and the immunizing viruses, the more decrease in HAI. But again, the decrease is faster for H3N2 than H1N1 because, as we've just seen, has more mutations and more amino acid changes in it. Now they wonder, maybe there are certain mutations, amino acid changes that say affect glycosylation, because we know that if there's a sugar group, it can occlude antibody binding. And if you take away the amino acid, it changes. So there's no longer a sugar group, it won't block it. They also have this other thing called cluster transition. I tried to look it up. I have no idea what that means. So we'll just focus on the glycosylation. So even we don't know everything. We don't know anything, actually.
Now I'm not even close.
No, not at all. I mean, I think all of these are really just a question of, is it a unique feature of the hemagglutinin and the neuraminidase, or is it just time?
Yeah. Well, anyway, they look at these amino acid changes that would affect glycosylation, and they do find some effect on the cross-reactive titles. So that could be playing a role as well, besides the nature of the amino acid itself. So far, everything has been done in ferrets. Is that a weasel, Alan?
It is a weasel.
So what's the taxonomy level of weasel?
They're carnivora.
It's just a colloquial name, right? Weasel.
Yeah. Okay.
So it's in mustelids.
Mustelids, yes. It's
right.
a genus.
Mustelid's a genus.
Genus Mustela.
So all mustelids are weasels?
Yes.
I see. And then
And
ferrets
all weasels are
some...
mustelids. And
Right.
they range from like the little short-tailed weasels that we get around here that are the size of squirrels up to badgers.
So I was going to say mustelid, you can sort of say is Latin for weasel, but it includes weasels, badgers, otters, polecats, martens, grissons, and wolverines.
Yeah.
Wow.
Those are all weasels.
Love it. I just heard you say weasel. That's why it set me off, Alan.
No, and ferrets are definitely weasels.
And ferrets are definitely not people.
No. But they're a pretty good model for flu, for some types of flu experiments.
Yeah, as you'll see, that they actually add to that here. So they want to compare humans and ferrets in terms of this breadth of antibody responses, right, that we've been looking at in ferrets. And so they have data from humans, from 6 children aged 9 to 24 months, 138 titrations, and 23 tested viruses. So the whole setup is the same as in the ferrets, but this is from young kids, and they're much fewer numbers.
I really like that they included this because it made me think about the challenge of finding humans who only had one exposure.
Yes.
Yes.
So these kids all had PCR-confirmed H3N2 infection. And they say, we assume that these antisera reflect cross-reactivity arising from a single infection because their children were probably too young to have a lot of episodes of infection with different H3N2 viruses, right? They're only 2 years old at the most. So
yeah, and just amplifying on Brianne's point there, I mean, not only do you have to find infants, basically, so they've not been through more than, at most, 2 flu seasons. You also have to find infants that are old enough that you're not going to be just reading maternal antibodies.
Right.
And that
Mhm.
have that actually had flu, and you know what flu they got, so you've got to get PCR confirmed cases, which is probably why they're only looking at 6 of them.
Yeah, but
That's
I
why
just think
they-
about, people often will be like, oh, well, we really need to look in humans, not just in ferrets. And
Yeah.
it's important to think about the differences in the model and the number of infections and things like that, not just the difference in the organisms.
That's why they'd start with 9-month kids, because that's the maternal antibodies are largely gone by then, right?
Yep.
Okay. So basically, they find the same thing as for ferrets. They get these bell-shaped patterns. Remember, they're just looking at H3N2 here, which has that nice tight bell shape, and they overlap between the human and the single exposed ferrets. Okay,
Thus
so they're
proving that kids are weasels. No, sorry, that's not what it proves.
I know some adults who are weasels.
Yes.
Some kids are kind of wiggly.
Yeah.
At that age, though, that they haven't yet had the opportunity to become weaselly, right?
No, not
2 years.
particularly.
2 years. All right, so then they got brave based on these data, and they decided to try kids 3 years and older, 1,111 individuals, unvaccinated and probably encountering multiple antigenic clusters. So here now, you don't have a single H3N2 infection. You probably have multiple. But they do know they have H3N2 infections. And so for viruses with likely minimal and no exposure probability, that's before 4 years of age. So some of these were of that age. The HAI titers decrease with early isolation from the reference year, so as we saw in the ferrets. And then when you get older, you get a broader range of cross-reactivity for 4 years and older because they've had infection with different viruses that have been circulating. So it's not just that one virus, you've had multiple. So the cross-reactivity curve broadens, right? But it's still consistent with what they have been finding. So this is cool because we can conclude that similar things are likely happening in people. They want to know, well, what is the impact of getting infected over and over again with H3N2 on the breadth of antibodies that they can detect. So they looked at cross-reactivity of the immune response from a person's antibodies over the course of their lifetime. And after they've been first infected with H3N2 or vaccinated, and then what happens afterwards. And what they find, so they do this, these are data of the same sort that we've been talking about, HAI assays. What they find, I had to read this about 6 times before I understood this. Fold changes in the antibody landscape, so getting away from that original H3N2, were higher for viruses within the cross-reactivity breadth compared with those beyond this range, which is basically what we've been saying so far in this paper, right? The further you get away, the less cross-reactivity that you have.
So basically you have a fair amount of cross-reactivity within that curve.
Yes.
But not any cross-reactivity outside the curve.
That's right. So there's basically there's a minimal effect of repeated H3N2 infections on these HAI titers for viruses that are outside the cross-reactivity because they're too different, right?
You're going to get a lot of reactivity against that same area in the curve.
Right.
You're not going to just get broader and broader and broader and broader and broader and broader
Yeah.
antibody responses. And that's kind of a good thing because if you just got antibody responses that got broader over your whole life, then you would start to get things like autoimmune responses.
You would eventually be immune to everything, including yourself.
Yeah.
Right. So this is important for vaccination because it means as these viruses change, you have to keep up with them. You can't depend on that original infection or vaccination because after a certain point, so many years, depending on whether it's H3N2 or H1N1, no longer going to help you, right? Well, that's another question. We're looking at HAI titers, and we know that there's a correlation between HAI titers and susceptibility to severe influenza. So that's the big assumption you make here. But people have done this for many years, so they do have a pretty good handle on the correlates of protection. So these repeated exposures are not making major changes in your antibody breath. So that's the paper. Basically, there's this intrinsic association between your antibody responses and different isolation times because the virus is changing over time. So that makes sense. H3N2 has a limited antibody breath than H1N1, so the population immunity declines faster, and that's confirmed with population immunity studies. So, and then of course, this evolutionary rate doesn't fully explain the breadth of the cross-reactivity, as we said, right? You have to also look at, well, first of all, there's subtype-specific differences, H3N2 versus H1N1, and we don't actually know why that would be. But there's also something having to do with the isolation time that we don't know the explanation for. But they do say we probably need annual vaccine updates to minimize these mismatches. All right, so here's a cool part. I like this very much. Let's see if I can explain it. So they say in ferrets and young children, they say that this supports ferrets as a good representation of single exposure human model because the ferret data and the baby data looked similar. But then when you look in the older kids, you get asymmetric antibody breath. And so they say as individuals encounter antigenically different viruses, their antibody landscape expands preferentially toward contemporary viruses, while responses to unexposed or distant viruses beyond the 6-year cross-reactivity extent remain minimal, implying that back boosting may occur primarily, the word that we don't like, within the cross-reactivity range. But here's the cool part. They say our findings support progression from original antigenic sin, where the immune system preferentially recalls responses to the first strain, to antigenic seniority.
Yes.
Which introduces a hierarchy of stronger responses to earlier exposures. A hierarchy. I like this antigenic seniority.
Yeah.
You like that, Brianne?
Yeah, I'll take that.
Goes from original antigenic sin to antigenic seniority.
It's not just that, it's not absolutely that you care about the most senior or the first one, but you sort of care about the most senior one in your little group.
That's
Yes.
right. That's right. I thought that was cool. And also they note that if you're born in 1995 or 2004, you have similar antibody breath. So it doesn't matter when you were born, you get the same result, comparable protection after that first infection. So there you go. I thought that was interesting.
Very.
And informative about this breath. It shows you what you can do by just mining existing data. It's great. Nothing like a computer, right?
Right.
Okay, we have some time to do a couple of emails. Brianne, can you take that first one?
Sure. Anthony writes, he's quoting from an interview. The interviewer says, "Why should it be the artist rather than the scientist who perceives these relationships and foresees these trends?" And the interviewee, McLuhan, notes, because inherent in the artist's creative inspiration is the process of subliminally singing out environmental change. It's always been the artist who perceives the alterations in man caused by a new medium, who recognizes that the future is the present and uses his work to prepare the ground for it. But most people, from truck drivers to the literary Brahmins, are still blissfully ignorant of what the media do. And this is from an interview of Marshall McLuhan in 1969, who was a professor at the University of Toronto. And this is what he has said about how artists perceive the world.
This was interesting. Artists always have a different view of things, and I like it. Yeah. Okay, I'm just putting a new one in at the end about this removal of RFK Jr. Okay. All right, Alan, can you take the next one?
All right. Blue Pilgrim writes, I suspect that Trump is not nearly as ideologically or religiously attached to anti-vaccination as RFK Jr. or some other zealots, but is engaged more in transactional populism, and as such will be more vulnerable to popular pressure than as many people, including Republicans and more traditional conservatives, start to panic about the repression of vaccines and science. I suspect he will be the weak point in the struggle, and that organizing letter campaigns and such directed at Trump would give the best chances to replace Kennedy with someone who is gung-ho maha, make America healthy again, but less fanatical and anti-vax. Trump may not be educable about science, but he can feel which way the wind is blowing. If he decides to fire RFK Jr., he can do it, and he can find an excuse for it that will not destroy support from the ignorants and crazies at his base. Okay, interesting theory.
I just don't know how he thinks about things, but yeah, it's an interesting idea.
Yeah, I don't know how much of this is just what Trump thinks or what those around him think, and how many of them would be easy to convince on this front.
Yeah. And there's also the risk, I mean, if you're talking about lobbying to Trump himself, he has the oppositional traits of a 2-year-old. So if you tell him he made a bad decision in appointing RFK Jr., he will double down and standing behind RFK Jr. just because you said it was a bad idea.
On the other hand, if there are lots of deaths from infectious diseases in the coming years, he may decide that that's not acceptable, right? Because
depends on who's dying.
Yes. It's a clear difference from previous years. Right now, for many diseases, we don't have a lot of deaths. And if it changes, I think that could make an impact.
Yeah, the question is sort of when will that change happen, and how does it get linked to what's happening now?
Yeah, it's hard to link it with RFK, right? But you say, look, you made these, you eliminated these vaccines, and now people are dying of these illnesses that are vaccine preventable. A good lawyer could take it, right?
Yes.
Jay writes, TWiV. I ran into a great quotation this morning. I shared it with my office staff. This is from David Ho in his 1998 MIT commencement address. When the wonder of nature is revealed, one is left breathless and awestruck. It gives a link for that if you want to hear the whole address. Thank you for your incredible work, Jay, as a chief medical officer at a clinic in Los Angeles. Thank you, Jay. Yes, nature is amazing. It's also not kind, Nature is not human-hearted. That's a quote by Lao Tse-tung, I think. Okay, we're back to Brianne.
Massimo writes, Dear TWiV crew, it was a sunny day in South Florida in mid-April 2020. My lab had just set up a highly sensitive SARS-CoV-2 testing protocol. Most COVID restrictions were still in place, but were not going to last much longer. It's Florida, after all. We don't slow down for viruses. And I was catching up on old TWiV episodes on a long walk to work. TWiV 531 had an interesting title, circ-du-RNA, and it was about some odd circular RNAs being generated by some herpes viruses via a mechanism of backsplicing, where a donor splice site is not spliced to a downstream acceptor site, but to an upstream one, forming a circle. For over 20 years, I had tried to figure out how the HIV primary transcript is transcribed and spliced into several shorter mRNAs. Could it be that HIV messengers are backspliced into circular RNAs? I got to the lab, I started looking at the genome maps, and started drawing circles. I counted 15 of them and called one of my students over to discuss a long and convoluted plan to figure out if HIV was making circles, and if so, what was their function. 5 years later, our first work on HIV circRNAs is out, and he gives a link to a paper in Nature. There are indeed at least 15 circRNAs generated by backsplicing of the viral genome, and we started slowly figuring out mechanisms and functions for all of them. Thanks to a long walk and an old TWiV episode, now we know a bit more about a virus and its host. Keep informing and inspiring, and Massimo is a professor at the Charles Schmidt College of Medicine at Florida Atlantic University.
Nice.
to RNA. I like the title, Alan.
Yeah.
Be good. I'm happy when people get some kind of influence from us.
Mm-hmm.
Listen to TWiV, get more publications.
Alan, you're next.
Anne writes, "Hello, Professor Racaniello. Carl Zimmer is on the latest episode of Radiolab about Thermus aquaticus, and in his brief appearance mentions all of the research being cut. Here's a link to his newsletter, which provides more information. Seeing the cuts in print makes me sad and angry. So much damage is being done. Maybe it would be worthwhile to have Zimmer on TWiV to talk more about these cuts. The science community must have mobilization efforts underway to rally support for this research. If you or your listeners know about them, the rest of us can help get the word out." And sends a link to the interview. Regards, Anne. P.S. Radiolab also did a similar segment with Veritasium about how PCR was developed and how TAC was instrumental in its success. I wasn't aware of Carrie Mullis nor his background, and provides a YouTube link to that.
He's a character. He was
Interesting character.
Carrie, and he ended up being a HIV denier, right?
Yep.
in the
Yes. Yes.
end. I mean, go figure.
Just goes to show a Nobel Prize doesn't prove a lot.
No, you cannot say this person is a Nobel Prize winner, therefore listen to what they say. No.
No.
Oh my gosh. Marjan writes, "Hello, are there studies on efficacy of, I think it's IPV.
Inactivated IPV, I think, yeah.
IPV, if given subcutaneously instead of intramuscularly. Wondering because of discussion a while back regarding oral polio vaccines being given due to lack of skilled staff to give IM shots." This is a good question. So it turns out that microneedle patches, which is kind of sub-Q, have been examined for many years. So these are little squares of plastic with tiny needles in them, and you saturate it with vaccine, and then you Band-Aid it to your skin, and the needles go into the dermis, and they immunize you. And for some pathogens, they work very well. But for poliovirus, they could put IPV on these microneedle patches, and you get a less than ideal immune response. And then, despite our insistence that people do not like needles, in lower income countries, they trust needles, not microneedle patches. But in the end, it doesn't address the gut immunity problem, right? You're not immunizing the gut, and that may be important. So there you go, Marjan. It's a good question, but I don't think these patches are going to work for poliovirus. And one more from Brianne.
Jennifer writes, "We are encouraging folks to contact their federal elected officials and demand RFK Jr.'s removal from office." Jennifer gives a link to a place to contact your elected officials. "If you can help share this among your listeners and followers, it would be appreciated. If it's okay with you, I'd like to look through the timestamps of this year's TWiV episodes and share clips of when y'all discuss Kennedy's dangerous actions. If there's anything we can do together, let me know. Thanks for all you do to educate the public. Jen."
So let's see what we have here. Probably a similar thing. Yes, call to action. So you fill out this form, and then there's a letter that you can have sent to your elected official. R.F. Kennedy must be resign or be impeached. He has not kept the promises he made to senators during the confirmation process. I'm calling on you to take action to protect the health of my family and all the families you represent. So the fact that he didn't do what he said he would do during confirmation, it probably is not actionable, right?
Well, I mean, he flat out lied.
He did.
Yeah.
But in this administration, that's good. Right? It's
But if there's
considered-
enough political pressure, I mean, Congress does actually have the tools to stop this. The framers of the Constitution did consider the possibility of a demagogue taking over the country and trying to destroy democracy. What they did not consider was the possibility that there would be an entire major political party supporting that demagogue's actions and therefore neutering the effect of the other two branches of government.
Yeah, I guess they should have taken that into account, right?
Well, it would have been pretty hard to anticipate because they were assuming people would not be quite that stupid.
Yeah, we're in an era of stupidity for sure.
Yeah.
Well, anyway, Jennifer, we will, listeners, do this. Write this letter to your representatives and senators and
Yeah.
see if we can do something to get rid of them. The more noise you make, the better. If you make noise, they will listen. Enough of you make noise, they will listen. So thank you, Jennifer, for doing that. Okay, folks, it's now that time of TWiV that you all love, our picks of the week. Brianne, what do you have for us?
I have an article on CNN that I really enjoyed reading. So this is about a planetarium show at the American Museum of Natural History. I am very much aware of this planetarium show. They had one that I really liked that closed a little more than a week ago. And they closed this one that I really loved to open a new show, which I have not yet seen, as I was not yet, I haven't had time to go. And it turns out that when making this new planetarium show, they were using a bunch of data that had been collected about the Milky Way, but that had not been used for 3D visualization before. And they used it to make a 3D visualization of the Milky Way for this planetarium show so that you could look like you were in the Milky Way. And when they did that, they looked at some structures and they said, that's not the shape that we always draw that structure as. What's wrong with this? That shouldn't be there. They actually went back to the scientists who had collected the data and talked to them about it. And in the end, they actually discovered that the Oort cloud that is around our solar system is shaped in a different way than we ever thought. Because no one had ever thought to visualize it in a 3D representation the way they happen to have done for this planetarium show. And they now sort of have realized that this Oort cloud is elliptical and not spherical. Spiral and not spherical. So I think that gives them, interesting ideas about how our solar system is forming and how different things are forming in the solar system. But I also think it sort of shows some really cool reasons why it's not, we don't, just think that these planetarium shows are cool things for me to go see on a weekend. But they are, in fact, giving us new science and new knowledge because they're making us present things in different ways. Just like when we as TWiV are trying to present things for our audience, sometimes we, figure something out that we might not have thought of before. So I just thought this was such a great story. I was so excited to read this. And now I'm even more excited to go see this show than I was before.
This is awesome. This is just really, really awesome. I love it.
Yeah.
And it's a great example of how science can work sometimes. Yeah, I love it. It's good. Alan, what do you have for us?
I have a pick that I'm pretty sure was one of Brianne's picks years ago. And when she picked it, I put it on my list of books to read, which is a long list. And it often takes me quite a few years to get around to any particular. I've got books on my list that, they've been around for a while. So I finally got around to reading this one a few months ago, and now I moved it. As soon as I finished it, I put it on my TWiV picks list, which is another one that I eventually get to. This is Project Hail Mary by Andy Weir. If you read or saw the movie The Martian, that was Andy Weir's story. This is his subsequent novel, totally different subject. It's not on Mars. I am not going to, if you haven't read this and you're listening to this show, you need to read this story. It's just really, really well done. And, it's the main character is a science teacher who ends up in a situation that is just mind-boggling and figures his way out of it, as would a science teacher. And it's just, it's a really fun read. And it's good, hard sci-fi that is just thoroughly entertaining.
As soon as I saw this pick, when I was looking at the document, I smiled so big and I was so excited and just started thinking about how much I love this book. So yes, please read this book if you have not.
Yeah.
So how does it compare to his other two?
I find it much better.
Yeah.
So he has one called Artemis, which is okay. I like The Martian, although The Martian can get a little tech heavy sometimes. I think that Project Hail Mary is one that makes me think about things in new ways, in a lot of fun ways.
I have not read Artemis, but this is definitely the best of the two works of his that I do know.
Yeah, I saw him talk at an ASM meeting once. He gave a talk and then he sat down and answered. Actually, he just sat down and answered questions. He was interviewed by Kate Rubins. And,
Nice.
And I think also, I can't remember his name, the science writer who's very popular, young guy.
Ed Yong?
Ed Yong, yeah.
Ed Yong.
It was fun. He's a very normal guy, People would ask him questions about the microbiology of his books. He said, I don't know. You guys are the microbiologists. You answer it. You answer that question.
Yeah.
That's cool.
Yeah, highly recommend this book.
Yeah.
I second your pick, Adam. Or Alan, sorry.
Yeah, so if you, like me, have not gotten around to reading it yet, go ahead and do so.
I just finished reading Zen and the Art of Motorcycle Maintenance.
Oh, fun.
Which was a pick of Rich's over a year ago, and I finally finished it. And I read the afterword, and his motorcycle is in the Smithsonian, apparently. That's very cool.
I read that back in college. That's a classic.
My pick is a Nature article, which I believe is open access.
Nope.
It's not?
Nope.
Too bad.
That's sad.
At least, well, okay, so your link to it goes to Columbia University login.
Oh, that's
Let
no
me
good.
just try
I put
searching
the wrong.
Yeah,
that's
the
bad.
title, see if I can go in the front door at Nature. Here we go.
Yeah.
Yes. Yeah, I thought Nature's news articles were generally.
I have to find
Fix
another
the link. There we go. I fixed your link.
Thank you very much. Yeah, I use my Columbia Access, and then I accidentally often copy the URL from it, which is not good. Anyway, so it is open, right?
Yeah, you can. I just I've not logged in. Yeah, I'm not logged into the Nature site at the moment.
Okay.
But
anyway, it's a good article by Helen Pearson, How to Speak to a Vaccine Skeptic. Research reveals what works. So they essentially, there's a lot of research in anti-vaccine sentiment going on. Oh, God, this picture they have here, it just bothers me. People protest in 2019 to exempt children from a vaccine requirement. And this lady has this poster, vaccines, the more you know, the more you know. N-O. It just bugs me. She's smiling away, and she's wrong. So how do you tell her that? Well, you can't tell her she's a dumbass that doesn't know what they're talking about. That doesn't help because she'll just get pissed at you. So this article gives you some clues. And like the first thing you do if someone expresses uncertainty about vaccines, do not judge or dismiss them. You're a dumbass. You don't know what you're talking about. You don't think that works? No, it doesn't work.
no, that doesn't work.
May make you feel better, but it doesn't work, right? It just shuts them up and makes them more adamant. But there are a lot of reasons why people are hesitant. You should basically the idea is that you should sit down with them and have a calm conversation. They actually have good reasons why or reasons that are good to them, and it's not because they're all influenced by anti-vaxxers. And you should listen to them, especially if you're a scientist and you want to talk to a person who is not convinced about vaccines. You should ask them why they don't want to vaccinate their kids. And the WHO has put together a whole list of reasons parents don't want to vaccinate their kid. I don't trust safety and effectiveness. I don't trust the health system. I don't need it because the risks are low. I can't access it easily. So they have a database where you can go look at this. And they say also something very interesting, that blaming Kennedy for vaccine hesitancy ignores the problem that we've been having declined trust since COVID, which is probably true.
There's a graph here that
Yeah.
is sort of stunning because
Yeah.
it shows both the change before and after COVID, but it also just says the percent of people that say vaccines are important for children. And there are some countries on here that really shock me.
Yeah.
That I didn't realize they had that level of vaccine hesitancy. Japan, for example.
Japan,
I
yes.
just am shocked.
Very shocked about that. Yeah.
And by the way, I misspoke. This is one of these. It's not paywalled at the top articles, but after you get into it far enough that it's clear that you're really interested, then you hit the paywall.
Yeah, I know those. Those are
So about 1,000 words into the article, you will hit a wall.
Yeah. Anyway, there are a lot of good suggestions here if you're interested in trying to help people overcome their hesitancy. You should read it if you can get it. I think it was useful. So they conclude by saying scientists are light years behind anti-vax activists. These are highly organized, orchestrated, highly funded people trying to undermine vaccination as a program. It's very sad that, in my opinion, they ignore the data and they make up stuff. They make up misinformation. You know, they say HPV vaccines are killing people. It's wrong. It's not true. It's a lie. I don't understand. Well, they have no alternative. If they want to be anti-vax, they have to lie because there's no reason to be anti-vax. Bottom line.
For scientists, the most important thing to remember in a conversation like this is that, unlike us, a lot of people don't make decisions, and not that we're perfectly logical either. We're certainly not in a lot of areas of our lives, but people in general don't form beliefs around data. And so throwing data at people turns out to be a really bad strategy in a conversation like this. You're much better off focusing on finding out why they believe what they believe, meeting them where they are, and being transparent and honest about why you believe what you believe, and remembering that stories will carry the day. Anecdote in that context actually weighs more than data.
Listening and being sort of sympathetic and being a human being.
Yes.
Yes, which is what the administration is not being. They're not being human.
Well, yeah.
That too. As neither is RFK Jr. and all of them. Okay, enough of that. No, actually, it's not enough, but we have to move on. We have a listener pick from Jason. Hello, TWiViverse. 8C and windy here in Northeast PA, not far from TWiV's New York City world headquarters. It sounds like we're a big company, but this is just a couple of rooms and that's it. But thank you very much. I like that. I know how much you enjoy great photography, so when I saw this series on Twixter, I immediately thought of you. It's some of the most fascinating still and motion work I've ever seen in this area. Bonus, look for the video of immune cells attacking a virus. Thanks for everything you do, especially giving us a safe, smart port in the raging misinformation storm. That's great. I like that. So I'm looking, this is an X post. Is there a link? No.
Yeah. It looks like there's a thread, and at least right now I'm only getting the first post in the thread.
Yeah.
I would really like to see the rest of it. Right now I would like to see this immune cell that he mentions. And right now I just get this, as it says, terrifying photo of an ant's face. And it is kind of terrifying. I agree.
Yeah, I can't get in. I have to sign in.
Yeah, Zitter is paywalled now. So you have to, if you can look at this tweet, but as soon as you try to read more, it pings you.
What did you call it? Zitter?
Zitter, yes.
That's the new not mentioning the original name thing?
That's what, because it's Twitter to me, but now it's X apparently. So I'm just going to call it Zitter.
Yeah.
I don't like X either. I think that's dumb. I like Twitter a lot because it was all about tweeting, talking each other like birds tweeting, right? That was great. Okay, that'll do it for another TWiV. This is 1227. You can find the show notes at microbe.tv/TWiV. You can send us questions or comments or picks of the week to TWiV@microbe.tv. And if you enjoy these programs, please consider supporting us. Microbe.tv/contribute. Brian Barker is at Drew University Bioprof. Barker.bsky.social on Blue Sky. Thank you, Brienne.
Thanks. I learned a lot.
Alan Dove is at alandove.com. Turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at microbe.tv or on Blue Sky. microbetv.bsky.social. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene Ramsey for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.